Hello everyone, welcome back. Today we will discuss about charging of tRNA. Before proteins can be synthesized, amino acids must be accurately delivered to the ribosome. And this is where transfer RNA or tRNA plays a crucial role. However, tRNA molecules are not functional on their own. They must first be charged with the correct amino acid in a highly specific and energy-dependent process known as tRNA charging or aminoacylation. The step is carried out by a group of enzymes called aminoacyl-tRNA synthetases, which ensure that each tRNA is linked to its corresponding amino acid with remarkable precision. The accuracy of this process is critical because even a single mistake at this stage can lead to incorporation of the wrong amino acid, ultimately affecting the protein structure and function. In this presentation, we will explore how tRNA charging occurs, the enzymes involved, and why this step is essential for maintaining fidelity of protein synthesis. Adding an amino acid to tRNA is called aminoacylation or tRNA charging. An enzyme called aminoacyl-tRNA synthetase adds the correct amino acid to the corresponding tRNA. The process produces a charged tRNA or aminoacyl-tRNA. Now, if you look at the slide, this is the cloverleaf model of a tRNA molecule, and to its 3' end, all tRNAs will have CCA. This arm or the stem of the tRNA is known as the acceptor arm. Why this is called acceptor arm? Because it is this arm precisely to the 3' end of the tRNA, the amino acid is linked with the help of a ester bond, C double bond, COO. Okay. And this particular formation of covalent ester bond is done by the enzyme aminoacyl-tRNA synthetase. So eventually when we say a tRNA is charged, what we mean is an amino acid has been esterified to the 3' end of the tRNA molecule. tRNA charging uses ATP as a source of energy. Each amino acid has a specific aminoacyl transferase or aminoacyl-tRNA synthetase. The 3' nucleotides of tRNA is always CCA in all tRNAs. Remember, we have roughly around 41 tRNAs, and all tRNA, the 3' end will be always a CCA. And the amino acid binds to the ribose sugar of this linked to this adenine at the 2' carbon or the 3rd carbon of the ribose sugar. Okay, both are possible. Amino acid can be esterified to the 2nd carbon of the ribose sugar of adenine or 3rd carbon of the ribose sugar attached to the adenine at the 3' end of CCA in the tRNA. Now, this is what happens when the enzyme carries out this reaction. Amino acid is first activated using an ATP. At this step, amino acid reacts with ATP and it becomes aminoacyl adenylate because it gets attached to adenosine monophosphate or AMP. In this process, two phosphates are released as pyrophosphate. This release of pyrophosphate is very important in this case because as soon as pyrophosphate is released, as we already know in metabolism, that PPI has an autocatalytic activity. The moment it is released, it will get hydrolyzed, releasing a large amount of energy, which pulls this reaction towards the forward direction. Okay. So once amino acid adenylate has been formed or aminoacyl AMP has been formed, PPI will leave and this aminoacyl AMP is now ready to interact or react with the cognate tRNA. Cognate means corresponding tRNA. Okay. Now this aminoacyl AMP amino acid has, in presence of tRNA, has more affinity for tRNA than the AMP. Therefore, it eventually reacts with tRNA and then it becomes aminoacyl tRNA and AMP is released out. Okay. This is the whole reaction which takes place during the charging of tRNA. Now, on the surface of the enzyme, supposing you can imagine that this whole thing is the enzyme surface or active site. What is happening? Charging takes place in two steps. In the first step, enzyme, amino acid, and ATP. Okay, these are the three things which are interacting together. So enzyme has a binding site for amino acid, also for ATP. Okay, so first, at the active site of the enzyme, amino acid reacts with ATP, forms aminoacyl AMP, and PPI is released. Aminoacyl AMP is still bound to the active site of the enzyme. Okay, and PPI is released and eventually broken down, releasing large amount of energy, which stabilizes aminoacyl AMP. Okay, in the second step, this is the second step. In the second step, aminoacyl AMP can now react with tRNA, eventually forming aminoacyl tRNA plus AMP, which is released. Okay, so reaction takes place in two steps. The first step, amino acid is activated using ATP. In the second step, aminoacyl AMP reacts with cognate tRNA, which eventually releases aminoacyl tRNA and AMP. So this is what the reaction is. Now, so let's revise the process one more time. Aminoacyl tRNA synthesis is a two-step process, two-step reaction. In the first step, amino acid and ATP will react at this active site, forming aminoacyl AMP and PPI. Then, at step 2, aminoacyl AMP will react with tRNA, forming aminoacyl tRNA, and AMP will be released. Now, if you consider the sum total of these two reactions, what is happening is on the left-hand side, amino acid, ATP, and tRNA is left because this aminoacyl AMP is there at the left-hand side as well as the right-hand side. So this gets cut. Okay. So this is not there. This also get cancelled because both the things are the same thing. Okay. So what do we have now? Amino acid, ATP, and tRNA. And what are the products? Aminoacyl tRNA, AMP, and PPI. This is the overall reaction of charging of tRNA. So this is the actual reaction of aminoacyl tRNA synthetase, how it charges tRNA in two steps. The first step, amino acid reacts with ATP in presence of Mg2 plus and aminoacyl-tRNA synthetase enzyme. It creates aminoacyl AMP and PPI is released. Okay. In step 2, aminoacyl AMP reacts with tRNA and gives you aminoacyl-tRNA, also AMP as a byproduct. This is the overall reaction that takes place during charging. This is a simple representation of what is happening at the active site of the enzyme. Supposing this is the enzyme, it has three binding sites, 1, 2, and 3. In the first step, ATP binds the active site and the amino acid valine binds the active site. I have taken valine as a representative example. Okay. And then in presence of ATP, valine is activated and valine is converted into valine AMP. Okay. And PPI is released, which undergoes autocatalysis. Remember, whenever PPI is released, it will be hydrolyzed, pyrophosphatase enzyme, and or autocatalysis, and it will release large amount of energy, which will pull the reaction, which will stabilize valine AMP intermediate. Okay. Once this step is over, tRNA is bound at the tRNA binding site on the enzyme, and now this valine AMP is in close association with tRNA, and it finally reacts, forming valine tRNA, and AMP is released. Finally, when both the steps are over, enzyme regains its actual structure, releasing both the products, valine tRNA and AMP as the byproduct. So this is what is called charging of tRNA. So that's all. You must remember that charging is the most important step because even before the protein synthesis starts, all the tRNAs are priorly charged with their cognate amino acid. And this work is done by a group of enzymes very specific for their substrate, that is the amino acid and the tRNA. And this group of enzymes are called aminoacyl-tRNA synthetases. If the synthetase does a wrong acylation, there are chances that a wrong amino acid will be incorporated during protein synthesis. Therefore, the work of aminoacyl-tRNA synthetases and the step of charging is very crucial to create correct, accurate proteins during the process of translation. In the next video, we'll see you with another topic from molecular biology course. Thank you.